everyone, welcome back to the channel. In today's video, I'm going to cover the next part from the CSIR Net Life Science syllabus, Unit 1 Biochemistry, and today's topic is chemical bond and molecular interaction. I want you to please go through the first video so that it will be easy for you to understand this part. Without wasting any time, let's get started. Chemical bonds and molecular interaction, Unit 1. Starting with what are ionic bonds? As the name depicts, ions, when a positively charged ions, they come together with the help of the electrostatic attraction force, they will form the ionic bond. Now why this electrostatic attraction? Why it is given? Because as in the previous video I have told you about the octet rule, that every atom try to find stability in the environment, and for that stability it must complete its outer shell with 8 electrons. That's the octet rule. So in this case, the cation, that is a positive charging carrying ion, and anion, that is a negative charge carrying an ion, they will give or lose some electrons to form the ionic bond with the help of the force that is known as the electrostatic attraction force. It is commonly found in salt, as you can see in the diagram here. It's the lattice structure of the stable salt NaCl formed by the ionic bond, and it is also found in the biological system, including various proteins and the biological membrane. In protein, basically we call them salt bridges because they are not found between cation or anion type of properties, but they are present between the acidic and basic amino acid like aspartic acid, negative charge carrying, and basic amino acid like lysine. So here we call them salt bridges. The most important thing, their strength basically depends upon the medium. Why? I am going to tell you in the further slides. As you just remember that in vacuum it is stronger and in water it is weaker. Now question is why it is so. Let's get moving. Here you can see a general strength of this ionic bond is 20 to 80 kilojoule per mole. This force of attraction that is causing these ions to come together and form this ionic bond is inversely proportional to the distance square, that is the distance between these charges. And the water, it contains high dielectric constant. Now you must be confused, what is this dielectric constant? Just wait, I will explain everything. This high dielectric constant causes these ions to break down in water and not form easily these ionic bonds. So they have a weak tendency to form the ionic bond. The energy, how much energy you can say ki how much energy is required to form an ionic bond, you can find it by Coulomb's law. Now this is again, what is this? Just wait, I will explain these 2 points in the coming slide. In proteins, if you can see in the figure, it is a salt bridge in hemoglobin. You all know the hemoglobin structure, it contains alpha and beta chain. So tertiary structure are formed when different chain combine together and they form folded structure. For that particular structure, tertiary structure formation, you can see here these type of ionic bonds are formed which are known as salt bridges. So they are found in the proteins also. Now let's understand this dielectric constant part and the Coulomb's law. So what is this dielectric constant? It tells you about the ability of a medium to reduce the force between charges. If I simply say, in the diagram you can see the Na positive and Cl negative being separated. They are not able to form the ionic bond. Why? You can see they are surrounded by water molecules around them. Because these water molecules are basically polar in nature. You can see the polarity sign over there. And these polar nature try to come close to these ions and they form a hydration shell around these particular ions. So they are not easy to freely come close and form the ionic bond. That's why in medium, or specifically in medium such as water, which has a high dielectric constant property due to its polarity nature, there is a very less or weaker ionic bonds found. Now as I have told you, Coulomb's law is important for finding the energy, how much energy being consumed for the formation of these ionic bonds. It's nothing, it's just an equation for the proportionality. I have just told you that force of electrostatic force of attraction, because two positively charged ions are coming together, so that means they have attraction. Electrostatic attraction because they are not going to move itself, they are just giving or losing some electrons to complete the octet. That's why we call it as electrostatic. So when this type of attraction force happen, it is inversely proportional to the distance square. You can see here, we just equate that r square proportionality with the constant forms. You can see here, it's the product of the charges of the two ions divided by 4 pi. Eta is basically the dielectric constant and r square. So this whole equation will tell you how much amount of ionic bond strength or force is present there to make two opposite charge ions form these type of bonds. So it's just a thermodynamical property. You just need to remember that this is the type of force you can use to calculate the energy for the ionic bond. So this is the concept that why in water salts easily dissolve. The reason is salt has a dielectric constant, high dielectric constant. When you add the salt in a water, it will get dissociate ionic bond and easily get dissolved. So that's the concept behind it. I hope you understand about the ionic bond. Moving on to the next important bonds, that are the covalent bonds. They are just different. In ionic bond, you just know that the positively charged ions give or lose electrons to complete its octet. But in covalent bond, these atoms, we are not talking about ions here, we are talking about atoms here. We are not saying they have charges, we are just saying the atoms will share the electron pair. Remember, it's a pair, electron pair being shared between them. And because it's between atoms, they are just clubbing together, sharing the electrons in a same space. That means they are carrying strong high energy. That's why their bond strength is equal to 200 to 400 kilojoule per mole. Where they are found? They are found in basic biomolecules like DNA, proteins, and the simple example like carbon-carbon bond, carbon-hydrogen bond, peptide bond, phosphodiester bond. Name simple biomolecule and their interactions, their bond. Most of the bonds are covalent bonds because they are the strongest. And most of our elements, dominating elements, in the previous video I told you chenops. These elements show the property not of the ionic properties, but they are in the less ionic nature, so they are basically going for sharing of electrons. So that's why the dominating type of bonds in the biological system again are the covalent bonds. So they are the most stable and they are found in the biological macromolecules. So in the side diagram you can see here N terminal, C terminal of amino acid. You can see that the bond form, peptide bond is shown here, and when a peptide bond form, the water molecule release and these two amino acid form the peptide bond. This is a simple example of the covalent bond. What are the types of chemical bonds? It can be a single bond when a one pair of electron share, double bond, two pair of electron share, triple bond, three pair of electron share, etc. So basically triple bond, three types of bond are there. In the diagram just see, you can see the sharing of the electron in the last outermost shell denoted by the black dot. You can see here one pair, two pair and three pair. Now what is the bond strength here? As I have told you, covalent bond strength is very high, 200 to 400 kilojoule per mole. It basically related with the bond length but inversely. The more the bond length of the atoms, means the atoms when they are forming the bond, the more the coverage area for them. Like you and your friend are holding hands, the farther you are from each other, it's more difficult for both of you to hold those hands together, right? So it is same like that. If you are more close to one another, it will be more easy for you to hold hands. So that's the concept same. So more the bond length, it will be lesser the strength. So example, simple oxygen, nitrogen molecule, and in biological system, you know, the unsaturated fatty acids are there. So that's the concept here. About polar and non-polar covalent bonds, it is also important because most of questions come from this part. Covalent bonds are also divided on the basis of the polarity because not all molecules carry the charge. So the polar nature of the covalent bonds are then when the sharing of the electron is not equal. It is an unequal sharing. Why unequal sharing? When, if you just take example, one person is very strong, gym going person, and one is very weak a person. If they try to hold hand, it is definitely the stronger person that is going to pull the hand towards it with more force. So same in the molecules. So the more electronegative atom will have more electron sharing part. So the more electron cloud will be towards the electronegative atom than towards the electropositive atom. In the non-polar nature, there is the equal sharing. So the polarity of these two molecules is due to the electronegativity. There is no electronegativity difference. That's why they are sharing electrons equally. That's the difference between them. Now, why this polar and non-polar is important? You took the example of water in the dielectric constant, that was polar molecule. So it basically defines the solubility, reactivity, and molecular interaction. It's very important. So that's why polarity is very important. It explains why hydrogen bond and hydrophobic bonds or interaction form. We will see that in the next coming slides. So it's a universal solvent is water. Why? Because it's a polar in nature. So it basically dissolves everything. That was the basic part for the chemical bonds has completed. Now we are moving to the molecular interactions. These are the weak forces which are critical for biomolecular structure. There are 4 different type of the molecular interactions. These include ionic or electrostatic interaction, hydrogen bonds, van der Waals forces, hydrophobic interaction. They are weak individually, but when they are present in different multiple form, not just one type, when they multiple type and multiple number, they will form a strong collective bonds. So they govern the folding, stability, and recognition of the biomolecules. First, the electrostatic interactions. Their strength is 6 to 30 kilojoule per mole. They are weaker than covalent but stronger than the van der Waals forces. So they are of different types. You can see in the chart here. These are of 4 types. Charge, salt bridge type. We have already discussed, like ionic bonds, we just call them. It is type of salt bridge when we are talking in the biomolecule. There is attraction between two positive charge groups, like the negative charge acidic amino acid, positive amino acid, that is basic amino acid in the proteins. They form these salt bridges, just told you about that. So they help in stabilizing the tertiary quaternary structure and also found in the active sites. Now, charged dipole means one molecule is charged, ion, and the other molecule was not charged, but when it comes close during the sharing or bond formation, that time it will develop some polarity, partial charge on it. That partial charge we call it as a dipole. So it is, why it is called partial? Because it is only going to show that charge when in interaction with that opposite charge ion it was interaction. So when the ion get lost, it will again be a neutral atom. So that's why we call it a partial charge. Example here, sodium ion and water molecule. Water is polar in nature. It shows dipole polarity, but sodium is actually an ion. So that's why the solubility happen. Dipole, that means both of the molecules are coming together and developing partial charge. Individually, they are not carrying any charge. So it's happened when hydrogen bond form between water molecules, hydrogen and oxygen. So they are basically formed because of the polarity. And next is the induced dipole. Induced dipole means the nature of these molecules were non-polar. They are actually not charged, not polar. They are non-polar in nature, but they develop temporary dipole due to the nearby charge. Like chlorine ion, induced dipole in the nearby non-polar molecule. So in charged dipole and induced dipole, what's the difference there? In the above option, you can see that oxygen molecule was there, that is getting dipole. So the oxygen is actually a polar molecule in nature. It already has some capacity to develop the charge, polarity. But in non-polar molecule, it has very least capacity to develop any partial charge. So it's just temporary in nature. That's why we wrote it non-polar molecule. They are very weak and they just need for the stabilization of the macromolecule. What are the factors affecting the strength of these electrostatic interaction? In the diagram you can see the salt bridges found, the green color, you know what it is? It is the embedded protein, transmembrane protein present in the biological membrane, and in the extracellular part you can also see a red color protein. It is also again a protein and it is also carrying charge. So that is a periphery protein. So these type of salt bridges are found in nature, in biological system, in the different type of proteins and membranes. So let's understand the three factors which are going to make understand how these electrostatic interactions going to be formed. First is the distance, already told you, Coulomb's law. If you increase the distance, the force decline. Force decline means it is more difficult for them to form the electrostatic interaction. That's why salt bridges inside buried protein core, means they are in the less distance, the low dielectric environment, are stronger than those on the surface. If I simply tell you, you all know that proteins are present in the folded form when they are active. When they are not folded, that means they should be degraded, right? So in an active protein, when it is in the folded form, the inside present bond are stronger ionic bonds or stronger electrostatic interaction. Reason is the whole environment around it is not providing any dielectric constant nature, because it is whole the same non-polar nature inside the protein. So that's why it is easier for them to pull together and form salt bridges. But the periphery type of proteins, they are basically surrounded by water molecule, because our body is basically made of water. So that water will provide some dielectric constant, so weakening those salt bridges. That's why salt bridges are stronger in the core region of the protein. So again, this is the distance effect, and distance is basically due to the, you know, the Coulomb's law. Distance and dielectric constant both are inversely proportional to the force. Dielectric constant already told you. Next is salt concentration. If you add extra salt, if there is no extra charges to neutralize the charge, this will reduce the interaction, right? And high salt concentration can destabilize the protein by breaking salt bridges. If simply I say just salting out, you know, in a water, if you just dissolve more water, it will be a time after that there will be no salt dissolving, and it will start settling down. That is the salting out. Similarly, in our biological system, if you must know that in the proteins or any biomolecule, they have a balanced amount of different type of molecules and their interaction. If we increase the salt concentration over there, it is also going to give the same effect. There will be no neutralization of these opposite, because two opposite charge need to be equal in number and to neutralize each other. If any one increase, it will destabilize the bond, in simple words. Moving on to the next interaction, these are the van der Waals interaction. They are very weak in nature and they are not specific interaction. Means we are not going to call them, they are going to be specific, I am going to choose that atom or ion. No, they are not specific. Okay, they basically form when electron cloud fluctuate, creating a temporary dipole. I just told you that if two atoms just come close in this way, that they will form a attractive force between them. These van der Waals interaction form. They are not going to just wait for them to form any strong interaction. So that's why they are weak. They just depends upon the electron cloud distribution. So their strength is almost 4 kilojoule or less than that. Okay, per mole. Only effective at a very short distance. You can see here 0.3 to 0.4 nanometer is the shortest distance on which they are effective. When you are bringing these molecules too close, repulsive effect will happen due to the overlapping of the electron shell. That is explained by the Pauli exclusion principle. So if I just say, because these type of van der Waals interaction are between the non-polar molecules. Non-polar molecules don't carry any charge, they don't show any dipole. So that means they are not going to show any attraction by their own, right? But when the electron cloud is just, you know, a spark type of appear between them just by at certain distance, they will form a type of weak bond. But when you increase their distance, you will bring them together. Like you bring two balls together, they will burst out, because they individually have no polarity, have no dipole, so they have no intensity to form the any type of bond. They are just being getting there because of the spar. So by increasing the closeness, you are basically increasing the repulsion due to the overlapping of the electron shell. And when electron shell overlap, electron shell carry electrons. Electrons are of same charge, so they will cause repulsion. Same charge is repelled. This is the simple concept explained by the Pauli exclusion principle. Now for the example, in the enzyme substrate fit, that means the active site of an enzyme, most of the bonds are van der Waals. Now if there was a question once in the net exam only, which type of bonds are maximally found in the active site. Most of the people choose the option for the covalent bond, I guess. I am not sure, but they choose the covalent bond. Now what's wrong in here? If they form the covalent bond, covalent is the strongest bond, but for the enzyme it needs to work with different substrate at different times. So it just need the temporary bond to form between the substrate. So that's why the highly dominating bonds over the active sites in the enzymes are the van der Waals. Again, antigen antibody recognition. It is also a temporary type of interaction because they have the capacity to bind with different epitopes of the antigen. So that's why they need to be temporary bond. Protein folding, it's a cumulative effect. It means there are different type of bonds, but one of the bond is van der Waals interaction. Now the hydrogen bonds. Hydrogen bond is formed between hydrogen, as the name depicts. It is going to bound with one of the highly electronegative atom, and that high electronegative atom may be oxygen, may be nitrogen. Okay? Most of the time it is oxygen or nitrogen, but sometimes it can be another electronegative atom. It should be electronegative. Now because of the electronegativity, and hydrogen is just a polar molecule, that means this bond is always going to be directional in nature. It is always going to show its electron more towards the electronegative atom. So it's always directional. This is very important to remember. And it is partially covalent in nature because the other molecule is not highly electrostatic, that it just pull all the electrons. We are not talking about halogens here. We are just taking the molecules like oxygen and nitrogen, which also form the covalent bond, but it is not a proper covalent bond, it is just a partial covalent bond. Next, strength 12 to 30 kilojoule per mole. It is weaker than covalent bond, stronger than the van der Waals, and it is essential for DNA base pairing, protein secondary structure. You can see in the diagram, we know that hydrogen bond formed between A and T, adenine and thymine, guanine and cytosine form triple hydrogen bond. So these are the basic backbone stabilizing bonds. So hydrogen bonds are weak individually, but critical in biology. They are strong enough to stabilize DNA and protein, yet weak enough to break and reform quickly, giving life stability and flexibility. This is very important. They are very much weaker if individually we are talking about. Like in a water molecule, you know that water can easily form ice, but it can easily melt down if you just increase the temperature. That's the particular science over there for the hydrogen bond. If you see the character of a ice, it is so solid, you cannot break it, right? You will hurt if you just punch it. And in same while, if you just heat it, it will form steam and it will form gas. So that's why hydrogen bonds are individually weak, but when they collab together, they form solid structure like ice. And when they just breaking down, they will form first liquid, then in the gas. And same for the biological nature, they are easily to break down, but when they are present in multiple number, they are very strong to destroy. What are the factors affecting the strength of these hydrogen bonds? First is the angle. This is very important. You can see in the diagram below, the strong hydrogen bond is formed when the hydrogen bond formed between first atom and the other oxygen part having an angle of 180 degree. They are in the straight line. The two oxygen atoms or two electronegative atoms are in the straight line with that of the hydrogen. But in the other case, if the angle is less than 180 or more than 180, it will be weaker. It is always maximum at 180 because it will gives the maximum overlap, making the hydrogen bond stronger. Bent bonds are weaker because there will be crowdiness of electrons, so they will form somewhat repulsion over there. So straight will form proper overlapping, less repulsion. Next is the electronegativity of the donor or acceptor atom. So highly electronegative atoms like oxygen and nitrogen pull the electrons more, creating a stronger partial charges. That's the main reason hydrogen bonds are formed, because hydrogen itself is partial in nature, so it is easy for it to form a bond with a slightly electronegative atom. Now distance. The distance between the donor and acceptor must be close. It must be shorter than van der Waals contact for effective bond. Too far means weaker the bond. So it is also dependent on the distance. Less the distance, stronger the bond. Okay. The optimal geometry, stable hydrogen bond when angle, distance, atom type like donor and acceptor, one should be the donor, one should be the acceptor, and the hydrogen bond is strongest and stabilizes structures like DNA and protein. Now again you can see in the diagram the ROH and the other molecule is OP. Okay, so here the dash is representing the polarity, partial polarity, dipole, and OH forming a straight covalent bond. You understand this? And including these two, the blue color dash, these and this straight line, it is the complete hydrogen bond. So here one is the donor, it is completely giving the electrons, and one is the acceptor. So that's why here this is the hydrogen bond formation, and it is partially covalent, so it must show some partial covalent character. Special case and biological example of hydrogen bond, water versus H2S. If you just see H2S, H2O is chemically, you just know that they are similar, right? But there is a difference in oxygen and sulfur. Okay. Now water forms a stronger hydrogen bond in nature, so it is liquid at room temperature. But H2S form weak hydrogen bond, so it always stays as a gas. Now what's the reason here? It's the electronegativity difference. Okay, again the donor, which type of atom you are using. In DNA, base pair 2 hydrogen bond form between AT and 3GP. So already told you about this. In proteins, alpha helix, beta sheets are stabilized by these hydrogen bonds. You know that the type of hydrogen bond found between them depends, frame the type of secondary structure. It is alpha or beta. It depends upon how the hydrogen bond form between them. So remember this, hydrogen bond found in the secondary structure, and tertiary structure will always carry the ionic or salt bridges. Good. Now in the enzyme substrate recognition, multiple hydrogen bonds align substrates correctly inside active site, providing specificity. It's also important for the binding of the substrate and enzyme. These are the van der Waals bond. But for the accurate, you know that the theory about substrate and enzyme fit in theory. So the substrate only fit in the enzyme, that only it can form the bond. So to make the substrate fit along the enzyme active site, hydrogen bonds help. So this is the difference between the van der Waals and the hydrogen bond working in the enzyme substrate complex. Last are the hydrophobic interactions. They are non-polar molecules which aggregate in water. Just simply I am just telling you, to minimize the exposure to polar solvent. So what happened? I have told you that water has a high dielectric constant. So in thermodynamics we know that hydration shell of ordered water forms around non-polar group, clustering release water molecule, increasing entropy. What that means? If you know the thermodynamic rules say that there is always increase in entropy in the universe. In any reaction there is always going to be increase in entropy. So how to increase that entropy, that randomness? Okay, so when water molecule is there, the water molecules become more disordered when hydrophobe clusters together. Why? If you just see, if you remember the previous example of sodium chloride, water molecules surround in a particular angle towards a particular sodium ion, like 3 water molecules around. So it is very ordered type of structure arrangement, and in the whole solution it will form that stable structure, stable entropy. So entropy will definitely decrease over there because it is no randomness over there. So decrease in the randomness, decrease the entropy. So it just not going with the law of thermodynamics. So naturally in nature, because everything goes for the randomness to increase the entropy. So the non-polar molecules, because they are not going to carry any charge, they tend to come together and form hydrophobic interaction, which are also weak, so that water will not come in between them but just surround them, and it is more random in nature. Water will not form any particular angle or structure around them, it is just freely moving out in a random way. So that's why the hydrophobic interactions are formed. They are very natural in nature. In a simple way, when you add oil to the water, you always know, you just try to mix the oil, it will come together and form a single drop. That is the hydrophobic interactions. Okay. What are the factors affecting? So temperature, बिल्कुल, as you know, जब आप पानी को गर्म कर दो, उसमें ऑयल डाल दो, जैसे हम खाना बनाते हैं, तड़का लगाते हैं, तो उसमें ऑयल होता है, बट वो ऑयल सारा मिक्स हो जाता है, वो इकट्ठा तब होता है जब वो ठंडा हो जाता है. तो येस, टेम्परेचर एक्चुअली डिसरप्ट दीज इंट्रैक्शंस. चेन लेंथ, लॉन्गर द हाइड्रोकार्बन, स्ट्रॉन्गर द हाइड्रोफोबिक इफेक्ट, मीन्स हैवियर हो जाता है वो मॉलिक्यूल, ना हैवियर मॉलिक्यूल भी ईजी फॉर इट टू फॉर्म द क्लम्पिंग इफेक्ट टुगेदर. Shape, linear chain cluster more tightly than branched, because branched will provide angles for water to come and interact with them, but in a linear structure they are easy to fold up and not providing water to interact between them. Aromatic versus aliphatic, aromatic are less hydrophobic than long aliphatic, means straight chain. Again the same reason is there, not allowing water to enter between them. Salt or salting out, enhancing hydrophobic clustering, because when you add more salt, so salt will itself try to move water molecules towards itself, and the hydrophobic non-polar molecules are easier for them to get clumped together. Example, protein folding, again and again this is coming, and the membrane bilayer, phospholipid bilayer, you can see in the diagram, it is formed by the, you can see hydrophilic heads are present in the outer region, that is interacting with the water from the both sides, and between them are the hydrophobic tails. So lipid protein interaction is also one of the hydrophobic interaction. That's all for today. I hope you understand today's video. Thanks for watching.